In the previous video, we discussed how the amino acid composition of a peptide can have profound influence on the types of chemical and physical properties a peptide has. We saw that it can make peptides more soluble in water or more soluble in octane. It can influence whether a peptide has a net negative charge and therefore is an acidic peptide or positive charged and basic. In this video, we're going to explore the idea that it's not just what the amino acid composition is, but it's actually the order of amino acids within a polypeptide chain that can also influence the properties of that peptide. So up to this point, we've looked at the polypeptide chain as this linear construct of amino acids where the nitrogen, carbon, carbon backbone is kind of a straight line and the R groups come off of that. But as chemists, we know that we need to take into consideration the three-dimensional structure of central atoms, right? So within the polypeptide backbone, we have sp2 and sp3 hybridized atoms. And that means our bond angles are not 180 degrees, so the backbone is not linear. Instead, we end up with this kind of zigzagging pattern as we saw when we looked at the carbon chains of fatty acids. But in this case, we're alternating between carbons and nitrogens. And one of the most important aspects that comes out of this is the fact that our R groups, our side chains, are actually alternating. So our first side chain we see on the bottom of this peptide, but the second one is on top, the third one on bottom, the fourth one on top, and the fifth one on bottom. So we have this alternating orientation of the side chain. And that, just like amino acid composition, can very much influence the properties of a peptide. So let's go ahead and explore this idea and put it in the context of the physical properties of two different peptides. Okay? We will start off with a peptide composed of alanine, aspartic acid, lysine, valine, and alanine. Okay, so the way that I'm going to represent this peptide is with a straight line. And this straight line is meant to represent the zigzagging pattern that we observe within our polypeptide backbone. So anything that I draw above this line will be amino acids that occur on this side, and anything that I draw below this line will be side chains that occur on the other side. Okay, so we first have alanine, and alanine, as we've seen, is a CH3 group. Aspartic acid is a CH2, and then we have our carboxylate that's deprotonated under biological conditions. We have a lysine, which is 1, 2, 3, 4 carbons, and then an NH3 group, which is protonated under biological conditions, so it's a positive charge. We have valine, which is a carbon and then 2 methyl groups coming off. And finally, we have another alanine, which is a CH3. So for this peptide, what we observe is that we have a nonpolar group, we have a positive charge, and we have a nonpolar group on the bottom or on the bottom side of our polypeptide chain. On the top side, we have this negative charge and we have a nonpolar group. So in terms of the physical properties of each side, it kind of varies. We have some polar charged and we have some nonpolar on both sides. So let's see how this might change if we change the order of our polypeptide chain. So let's explore the polypeptide chain that has the order alanine, aspartic acid, valine, lysine, alanine. So when we compare this to the previous, all we see is a switching or a swapping of these two amino acid positions. Okay? So if we draw our polypeptide backbone again as a straight line, we have our alanine on bottom, so CH3. I have my aspartic acid, which we know is a carboxylate, so a negative charge. I have a valine, which was this branched chain amino acid, a lysine, which is 1, 2, 3, 4 carbons, and then our NH3. And finally, we have another alanine, which is a CH3. So now in this case, we see a nonpolar group, nonpolar, and nonpolar all on the same side, and we have our charged amino acids also on the same side. So this side of our polypeptide chain will interact well with water, whereas this side will interact well with octane or something that's nonpolar. So for this peptide, we can actually imagine it interacting quite nicely with the surface of a phospholipid or a lipid bilayer, because we have this polar region that can interact well with our polar head groups, right? So here are our polar head groups here, and we have water out on this side, and then we have our hydrophobic tails that would interact well with the non-polar hydrophobic regions of our peptide. So this peptide, as drawn, alanine, aspartic acid, valine, lysine, alanine, would actually very much like to be a peripheral membrane protein. Remember, these are proteins that sit on the surface of lipid bilayers. It can do this because it orients all of the non-polar side chains on one side of the peptide backbone and all the polar and charged side chains on the other. Whereas this first peptide we looked at, which had the same amino acid composition, but now we have a mixture of polar and non-polar amino acids on the same side, so this certainly would not favorably interact well with the surface of a phospholipid.